Ever wondered how we understand the genetic code of living organisms? Well, let's dive into the fascinating world of DNA, the blueprint of life. DNA, or deoxyribonucleic acid, is a complex molecule that carries the genetic instructions used in the growth, development, functioning, and reproduction of all known living organisms. Now, to unpack these instructions, we need to read the DNA, and that's where DNA sequencing comes into play. DNA sequencing refers to methods for determining the order of the nucleotide bases, adenine, guanine, cytosine, and thymine, in a molecule of DNA. Just like letters forming words in a sentence, these bases form the genetic code that instructs the formation and function of organisms. It's like deciphering a complex biological language. The journey of understanding this genetic language has been quite a ride. The first DNA sequence was obtained by academic researchers using laboratories methods based on two-dimensional chromatography in the early 1970s. And that's where our story of DNA sequencing begins. From primitive methods to high-tech solutions, DNA sequencing has come a long way. And what a journey it has been. The first DNA sequence was obtained through two-dimensional chromatography, a laborious and time-consuming process confined to the academic laboratories of the early 1970s. Fast forward a few years, and the development of dye-based sequencing methods revolutionized the field. These advanced techniques, paired with automated analysis, transformed DNA sequencing into a faster, more accessible process. The once painstaking task of determining the order of the nucleotide bases, adenine, guanine, cytosine, and thymine, in a molecule of DNA became a streamlined, efficient process. Yet, the story of DNA sequencing is not just about the technology, but also about the methods. Two main approaches have risen to prominence in this scientific saga: the chemical method, also known as the Maxim-Gilbert method, and the chain termination method, often referred to as the Sanger dideoxy method. The chemical method, named after its inventors, is a fascinating process that relies on the relative chemical liability of different nucleotide bonds. It's a method that has its roots in the chemistry of life itself. On the other hand, the chain termination method interrupts the elongation of DNA sequences by incorporating dideoxynucleotides into the sequences. This method, named after its inventor, is a testament to human ingenuity and our ability to manipulate the very building blocks of life. Each of these methods has its unique strengths and applications. The chemical method offers a deep dive into the chemical properties of DNA, while the chain termination method provides a fast, simple way to sequence DNA. But if we were to pick a crowd favorite, the chain termination method seems to have won the popularity contest in the scientific community. Its speed and simplicity have made it the go-to choice for many researchers in the field. While both methods have their merits, the chain termination method is more commonly used due to its speed and simplicity. And so the evolution of DNA sequencing continues, advancing our understanding of life one nucleotide at a time. With advanced equipment and artificial intelligence, DNA sequencing is now more efficient than ever. The introduction of artificial intelligence and advanced infrastructure in laboratories worldwide has revolutionized the process of DNA sequencing. These advancements have not only increased the speed and accuracy of sequencing, but also have made it more affordable and accessible. The impact of this revolution is far-reaching, especially in biological research. Today's researchers can sequence entire genomes in a matter of days, opening up new avenues for understanding the complexities of life. This efficiency has propelled us into the era of personalized medicine, where treatments can be tailored to an individual's genetic makeup. It has also allowed us to study the evolution of species, to track the spread of diseases, and even to solve criminal cases with forensic genetics. As we continue to decode the secrets of life, DNA sequencing remains a critical tool in our scientific arsenal.